Good morning, students. In this video, I shall discuss some basic concepts required to solve the problems related to Mendel's experiment. Now, in the examination, sometimes some problems related to the Mendel's experiments are given. So that is why the students may face problem at the time. So that is why before solving the problem or before going to solve the problem, the students must have the idea about this particular Mendel's experimental problem. So first thing, they must clearly understood that the concept of crossbreeding. Then what is crossbreeding? How the male plant and the female plant they cross, and what is the hybridization technique? How the emasculation and bagging procedure is adopted there. This process must be clearly understood by them so that they will not face problem in solving the problem. Now, what is P1 generation, or that is called parental generation? What is filial generation? F1, F2, F3, etc. So this generation, how the monohybrid, only one character is considered at a time, that is monohybrid cross experiment. What is dihybrid cross experiment? Two pairs of contrasting characters are considered at a time, then it is dihybrid cross experiment. Then how this test cross is done to know the genotype of the dominant parent. So that is why this concept must be clear so that they will not face problem in solving the problem. Now, for particularly the Mendel's experiment, they must have a clear idea about the 7 pairs of contrasting characters which Mendel actually used in his experiment. Now, these 7 pairs of contrasting characters of pea plant, and what are the phenotypes, right? A round and wrinkled, yellow and green, then whether the flower is purple or it is white. So these are the different phenotypes, and all the phenotypes, they have specific genotypes and the notation of the genotypes. For example, we are taking the capital T for tall and small t for dwarf. So this type of notation of the genotype must be clearly understood and must be practiced properly. Then which one is the dominant variety? Suppose you are tall and dwarf. Tall is the dominant trait and TT, that is the recessive trait, or that is your dwarf variety. So that is why this dominant recessive nature of the alleles of the pea plant, they must be clearly understood so that they will not face problem. Now next one, what are the homozygous condition? What are heterozygous condition? For example, capital T, it is the homozygous condition, homozygous dominant, and small t, these are homozygous. But sometimes the dominant and the recessive parent, they remain together. That is called the heterozygous condition. And it is in case of monohybrid. In case of dihybrid, R, Y, Y. So these are the heterozygous condition. So that is why this concept of heterozygous condition must be understood by the first problem. Now, what are the different process of gamete formation? Like gametes. The gametes are formed from the parent, or parental cells are formed by the process of meiosis. In the meiosis, the chromosome number, and like that, here the alleles are separated, and here the diploid parents, they form the haploid gamete. And how the capital, suppose the parent is having capital T genotype, then the gametes which are formed as capital T1 and another one. So these are the two gametes which are formed here. Either in case of male, it is the pollen grains, or in case of female, it is the egg cell. So these are formed there. So that is why this pattern of our process of gamete formation must be clearly understood so that they can easily draw the pattern and they can easily solve the problem. Now next one, the random and independent pattern of recombination of the male and female gamete to form the progeny. That means how this gamete, suppose this is the one gamete and this is another gamete, and how these two gametes fuse together to form a recombinant. So that is why here the random and independent pattern of recombination. So the male gamete and the female gamete, they normally fuse randomly and they are also independent to one another. So that is why this concept of random and independent fusion must be understood, and the pattern of recombination, right? So how the male gamete and the female gamete, they recombine together to form a new recombinant, and the male and female gametes normally form the progeny. So this progeny is having the genotype and the phenotype according to the fusion of the male and the female gamete. So this concept must be clear by the students so that they will not face problem there. Now, what is the genotype? Then what is the genotype of the progeny? Then what is the phenotype of the progeny? Suppose genotype, it is your capital T small t, then it will be the phenotype is the tall one. And if this is small t small t, then it is the dwarf one. So that is round, yellow, wrinkle like that. So that is why this genotype and phenotype must be clearly understood and mentioned. Then next one, that is the ratios of these genotypes and the phenotypes. In case of monohybrid cross experiment, the genotype ratio is 1:2:1, whereas the phenotype ratio is 3:1. And then what is this genotype ratio and what is this phenotype ratio? They must be understood clearly. In case of dihybrid cross experiment, it is 9:3:3:1. It is the phenotype ratio. But in case of dihybrid cross experiment, the genotype ratio is more complicated, of course, but sometimes they are asked from that person also. That is why they also know about the genotype or genotype ratio of the different progeny, and so that they will not face problem in solving the problem. And these new recombinants which are formed there in the form of, they must represent them in the form of Punnett square. So this Punnett square method is commonly used to solve the problem of the Mendel experiment. So that is why they must have an idea about this Punnett square and how the Punnett square is prepared and how in the Punnett square the different recombinants are placed and their genotype and phenotypes are determined. Now next one, they must have the idea about the Mendel's laws and the analysis of this Mendel's laws. The law of dominance, law of segregation, law of independent assessment, and what are the ratios of the monohybrid cross, what are the ratios of dihybrid cross, what is the ratio of test cross, that is your 1:1, and how the test cross is used or why the test cross is used in case of this Mendel experiment. So that is why these are the basic concepts one student must know so that they can easily solve the problem. Otherwise, they definitely face the problem. So it is my sincere request to all the students to please go through this concept, and they must clear their concept before going to solve the problems related to Mendel experiments.